Assalamualaikum and hi everyone. We are from Group 2. We will present a home-based project report SSCB 1613 Microbiology. We have 5 group members and I, Nur Nashita, we will present about the introduction. And next is Nur Nazira Ajana, we present about the objective and experimental setup. Muhammad Tafiq, we present about the results of this experiment. Nurul Laili will present about the discussion. And lastly is Tuan Sharifah Nawar, we present about the conclusion of this experiment. Contents of our report: first is introduction, second is objective, third is experimental setup, fourth is results, fifth is discussion, and lastly the conclusion of this experiment. Is Nunashita Biti Santro will present about the introduction of this experiment. As we know, microorganism is an organism that microscopic, which is too small to be seen by the naked eye. Microorganism is also an ubiquitous organism that we can find nearly everywhere around us. It can survive in every type of environment, as we can find them in water, air, soil, the body of plants and animals. We can also find them in human body, and especially we can find them in food. There are certain of microorganisms that can survive and live in extreme conditions, such as in environment with low or high temperature and too acidic or too alkaline environment. The most common microorganisms that we know are bacteria and fungi. In this experiment, we can observe the gradual growth of microorganisms on the food samples that we use. Usually, only yeast, mold, and bacteria are growing on most food. There are a few factors that influence the growth of microorganisms. Each microorganism has growth optimally at a particular temperature, pH, oxygen levels, and water activity. And different types of microorganisms require their own optimum environmental to grow. In this experiment, temperature has a massive effect to microbial growth. At high temperature, it can help speed up the growth of microbes, while at the low temperature, the growth of the microbes will become slower. But too high temperature can cause the loss of critical enzyme, and too low temperature can decrease enzyme activities. Microbes can be classified into 3 groups based on the optimum temperature for their growth. First is psychrophile. Psychrophile prefer temperature from 0 to 15 degrees Celsius for their growth. The optimum temperature for psychrophile to grow is at 15 degrees Celsius. Some microbes are psychrotolerant that grow at 20 to 30 degrees Celsius. They are responsible for most low temperature for food foliage. Second is mesophile. Mesophile has optimum growth between 25 to 45 degrees Celsius. Most of the bacteria are classified as mesophile. And lastly is thermophile. Thermophile has the optimum growth between 50 to 60 degrees Celsius, but extreme thermophile, known as hyperthermophile, has the optimum growth at 80 degrees Celsius. Based on the 3 groups of microorganisms, we can conclude that most of the food contamination occur at room temperature as bacteria grow rapidly between 4.4 degrees Celsius and 60 degrees Celsius, as many types of bacteria are classified as mesophile. Activity, known as AW, is a measure for the available amount of water in food that can use by microorganisms. Water activity is one of the factors that influence the microorganism growth in this experiment. Water activity of food will increase with temperature because when the temperature increase, amount of water vapor in food system also increase. Most food have a water activity above 0.95, and this value provides sufficient moisture for bacteria, yeast, and molds to grow on the food surface. At water activity value smaller than 0.6, almost no microorganism be able to grow because there is not enough water for microorganism use for their survival. As example, we can take is cracker that have low water activity. So as long as they are stored properly, they cannot be spoiled by microorganisms. While at water activity value below than 0.75, molds and yeast be able to grow where bacteria cannot grow. As example, we can take is peanut butter, jam, and honey. Therefore, we can conclude that water activity is one of important factor for microbial growth, as microorganisms need source of water to survive and grow. As we know, in this experiment, we need to further grow the microorganism that grows on the sample food. We can grow them use growth media such as nutrient agar, known as NA, and potato dextrose agar, known as PDA. PDA is used for the cultivation of fungi, especially yeast and molds that can be supplemented with acid to inhibit bacterial growth. NA is used for the growth of a wide variety of non-specific microorganisms because it contains many nutrients needed for the microbial growth. From the cultivation of microorganisms, we can obtain the pure culture of the microorganism that helps to identify if the types of microorganisms that grow are harmless or can cause any infections and diseases for ensure the safety of the food that we going to eat.
Hi, my name is Nazira Adriana binti Azlan, with metric number A21SC0290.
The objective of Microbiology Home-Based Project is to observe the microbial growth on different food samples and also prepare growth media for culturing microorganisms. For experiment 2.1, microbial growth food, 5 containers with lids and 5 containers without lids are washed to ensure it's clean and dry. Label each container and scrap paper with a permanent marker with the name of the sample and also starting date. The food material for this experiment were prepared such as rice, butter, milk, bread, and cracker. The container labeled as A are for closed system, whereas B for open system. After hands were washed and dry, the food material were placed in the container. Incubate all samples at room temperature. Perform the observation and data recording for 14 days at 24 hours interval. At the end of 14 days, keep the food sample from closed system for later use in section 2.3, which is media cultivation, and discard those from the open system. The second experiment is media preparation, DIY media. We have two, which is potato dextrose agar and also nutrient agar. For potato dextrose agar, 2 grams of potato are washed, and then peel off the skin and dice the potato. 250 ml of water were added and boil it for 30 minutes. After the potato is soft, collect the potato extract by sieving through cloth sieve. Adjust the potato extract to the volume 250 ml with water. 50 ml of water were heated and 5 grams of tartaric acid added, then mix it. The heating is turned off and leave it aside. The potato extract is reboiled and add 5 grams of sugar until it's dissolved. And then 5 grams of gelatin powder are added and continuously mix it to prevent the agar from sinking at the bottom. Next, the tartaric acid solution is added and keep mix it to avoid the solution from solidifying. After that, the hot media are transferred into the petri dishes and immediately close the container. For nutrient agar, 15 ml of water were heated and 5 grams of tartaric acid added, then mix it. And then, the heating is turned off and leave it aside. Next, 250 ml of water are boiled. 0.75 grams of beef stock powder were dissolved in boiling water. 1 gram of salt are added into the beef stock solution. 5 grams of gelatin powder are added and continuously mix it to prevent the agar from sinking at the bottom. The heat were adjusted to medium-low and bring the water to simmer. Tartaric acid solution is added into the beef stock solution and mix it to avoid the solution from solidifying. And then, the hot media are transferred into petri dishes and immediately close the container. The next experiment is media cultivation, which is a cultivation of microbes on solid media. First, an area where there is no and less traffic in the house are chosen to be a workbench. Adequate amount of alcohol plus sanitizer are sprayed on the surface of workbench. The alcohol is then wiped by using kitchen tissue paper. And then the candle is light up on the working area and leave it for 5 minutes. The candle flame is placed close and don't forget to wear mask. PDA and NA then are placed on the sterilized workbench. 1 plate of NA was taken. The sharp pointed knife is sterilized by flaming it on the flame of the candle. Sterile pointed knife is used to make a hole by cutting the agar in the center, and then the plate is closed immediately. Repeat the step for the rest of NA and PDA plates respectively. 1 container of food sample is taken. The sharp pointed knife is sterilized by flaming it on the flame of the candle. Observe the area where fungal growth visible and use the sterile pointed knife to cut a piece of the food sample with fungal growth. Using another hand, use a sterile needle to hold the food sample to cut it. The sample piece is removed using the sterile needle and transfer it into the NA and PDA hole. Close the plate by pressing back the lid. The steps are repeated for the rest of the food sample. For the sample that cannot be cut, which is milk, use the sterile nib tip to collect small amount of contaminated food sample and transfer it into NA and PDA hole respectively. All plates were then incubated in dark at room temperature. The petri dishes then are observed for 7 days at 24 hours interval.
Hi, Assalamualaikum. I'm Muhammad Taufik, and I will show you the experimental result from our group. For the bread sample in open system, there are no groups of microorganisms nor morphological characteristics observed from day 1 until day 6. However, the physical characteristics of the food started to change from day 3 when it becomes slightly hard, slightly dry, and still maintain the white color until day 6 it becomes slightly crunchy, dry, and white color. On day 7, several groups of microorganisms are observed. It starts with 2, and then at the end of the experiment, it stays with 4 groups of microorganisms with its respective morphological characteristics, which start with small grey and brown smudges on day 7, and at the end it becomes thicker with grey, brown, and white, grey, brown, white, and yellow color of smudges. For the physical characteristic of the food, it remains crunchy and dry until the end of the experiment. However, it starts to be covered with smudges on day 10, and starts to become slightly sour smell, starts to produce slightly sour smell from day 11 until day 14. As you can see in this picture, it starts with a clean white bread on day 3, and at the end it becomes fully covered with smudges on day 14. For the bread in closed system, no groups of microorganisms nor the morphological characteristics are observed on the bread. However, the physical characteristic of the bread slightly changes starting from day 3 when it appears to be slightly dry and then slightly hard until the end of the experiment. On day 11, few groups of microorganisms are observed with small grey dot on day 11, and the grey dot grows larger into small grey patches on day 12, and at the end it becomes a big grey and white patches and small fuzzy yellow dots on the bread samples. The physical characteristic of the bread remains hard and dry until day 14. In the picture you can see the difference from day 3 and day 14. Now for the plain cracker in open system, no differences are observed from starting until the end of the experiment. Only its physical characteristic changes, which it starts with crunchy and slightly oily, and then it becomes soft and dry until the end of the experiment. For the plain cracker in closed system, there is no groups of microorganisms as well, and the physical characteristic of the food remains the same from day 1 until day 14. The only observable changes on an open system of plain cracker, the mark of the oil surrounding the plain cracker increases from day 1 until day 14. For closed system, no differences are observed from day 1 until day 14. For the butter in open system, no obvious changes are recorded. The only difference is that its physical characteristic changes from solid and soft, and then on day 2 it becomes slightly melted until the end of the experiment. Similar observations are recorded on butter in a closed system where no groups of microorganisms or morphological characteristics are observed. However, the physical characteristic of the food is that it slightly melted starting from day 2 until the end of the experiment. As we can view from this picture, there are no changes from the beginning until the end of the experiment for the butter sample. For the rice sample in open system, there are a lot of changes observed, especially in the number of microorganisms and the morphological characteristics that occurs on the food sample. As you can view in this picture, on day 3, there are only small amount of orange mold on the rice sample, but at the end, which is on day 14, there are different types of microorganisms observed, such as green and black spores that grows large on the rice sample, orange molds, and small green spots on the rice sample. For the rice sample in a closed system, there are also a lot of changes that are observed and recorded. As you can see in this picture, on day 3, there are only a small number of yellow mold on the rice sample, but at day 14, it is covered with full black and dark colored mold with small and tiny spots of white microorganism on the sample. Now for the milk sample, the groups of the microorganism group and its morphological characteristics could not be observed for the milk sample. However, its physical characteristic changes starting from day 2, where it coagulated like curd and have sour smell from day 2 until day 14 of the experiment. Same goes with the milk sample in the closed system. The groups of microorganism and its morphological characteristics could not be observed in the milk sample. However, its physical characteristic changes from fresh white liquid and then it forms a suspended white solid on day 2 until the end of the experiment, and it starts to change from white solid into a yellowish solid from day 6 until day 8, and they produce bubbles on day 9 until the end of the experiment. These are the conditions of the milk sample on day 3 until day 14 in open system, and day 3 and day 14 in closed system. Next is the microbe sample incubation, where the samples are cultivated and incubated in potato dextrose agar plates and also neutron agar plates. So these are the observations recorded for the bread sample. This is a picture for PDA and NA plates from day 1 until day 7. This is the observation recorded for plain cracker, which there are no microbial activities observed. Same goes for butter. No microbial activities are observed on the butter sample. And for the rice samples, some microbial activities are observed, and the physical characteristic of the food changes. For the PDA, this is for the PDA agar plates, and this is for the neutron agar plates. Next is the milk sample in the potato dextrose agar plates, and finally these are the observation for neutron agar plates for the milk sample from day 1 until day 7.
Hello, my name is Nurulaili and I will present about discussion for this experiment. Good environment can make microbes successfully grow like in hotter or cold temperature. And they do best in warm, moist, protein-rich environment that is pH neutral or slightly acidic. Hereby, in this experiment, we require a room temperature for the microbes to grow on the food sample, which is bread, rice, biscuits, butter, and milk. First and second day for open system rice, there is no mold growing on the rice, but they're actually drying. This is due to the exposure of the air to the rice causing retrogradation of the starch. The loss of humidity of the rice in 48 hours, we can observe orange spots, molds on the rice. Orange moldy rice, it is rotten and has a bad smell. Spores will germinate forming thread-like cell called hyphae. And as for the closed system rice, we can see water vapor trapped inside the container, which is the condensation process. But within 2 hours of incubation, the rice starts to smell sour due to the fermentation process. And the fermentation process make the water vapor increase day by day. Because the bacteria spores grow and produce toxin, which is formation of lactic acid, which because of the lactic acid, the formation of the lactic acid, the fermentation process occur and make the rice smell sour. At 72 hours, pale yellow mold made the first show, and the mold's name is Aspergillus oryzin. Next, for open system bread, It becomes harder after 72 hours, being exposed to the air as the structure of the starchy carbohydrates start to crystallize. On 168 hours of incubation, some brown smudges molds showed up and a hint of small grey patches mold which is penicillium. From this experiment, all of the common molds were successfully observed. For example, the common molds that we can find from are penicillium, Rhizopus stolonifera, Cladosporium species, and Aspergillus species. For the closed system bread, there is nothing happened within 2 days of incubation, yet on the 3rd day, the bread started to harden and dry. The open system bread obtained more molds to closed system bread. So here we believe that molds also can grow because of the food was exposed to the air, as referred to the e-book said that the mold can grow from tiny spores that float around the air. When some of these spores fall onto a piece of damp food or other materials, they grow into molds. Then open system milk for open system milk, the milk coagulated like curd and smells sour within 48 hours until day 14. From Osman recommend anti-fat was a glitter that spoilage of milk and milk product result from growth of fermentative bacteria when storage temperatures are sufficiently high for psychotroph. Then for the closed system milk, it changed within 48 hours in room temperature, formed suspended white solid. Actually, all milk contain bacteria. The bacteria eat natural sugar in milk called digest lactose, which lactose is a disaccharide. As they digest lactose, a number of byproducts are created, including lactic acid. Then the pH will drop and casein molecule begin to clump and result the sour smell of milk. Within 240 hours of forming, within 240 hours, the milk forming bubbles, which the milk also release gas. No molds grew for both open and closed system plain cracker. For the open system, the plain cracker just becomes soft, and for closed system, they just appear fresh like the first day of the incubation. So for the butter, for both open system, butter just melted throughout the 14 days. Due to the high fat and low water content, the mold is not easily can grow. So for the cultivation in potato, citrus agar and nutrient agar, we need to cultivate the molds from all the ecosystem samples into the PDA, then store them in dark place and observe the growing molds inside the agar. We choose Rhizopus species to cultivate inside the PDA and NA for rice molds. The Rhizopus species able to grow faster inside the NA compared to the PDA. Rhizopus species covered more area in NA to PDA, however, refer to the ebook by Helicono and his colleague, the Rhizopus species should grow faster in PDA. The formula, there are two different molds were obtained in PDA and NA, where for PDA is dark spore, which also the Rhizopus, and in NA is white spores, which is Penicillium species. This Penicillium species is common mold that grow in cheese and other dairy products, include milk. These molds trigger the formation of mycotoxin. This natural toxin produced due to the fermentation process in dairy product. So if you swallow the mycotoxin, some disease happen, which acute toxicity includes gastrointestinal symptoms like vomiting and diarrhea, as well as acute liver disease. However, mycotoxin also can benefit human by their use as antibiotics, which is penicillin, immunosuppressants, which is cyclosporine, and in control of postpartum Homer H and migraine headache, which is ergot alkaloids. For the cultivation for NPDA, we took white spot mold, which is fusarium, and put it at the hole of in the hole of PDA. On the second day, a yellow mold covered with Rhizopus grew nearby the white mold that we cultivate. While we obtained a lot of small white spots scattered in the cultivation of bread, in NA also cultivation of bread. While in NA, also some Rhizopus species. Lastly, there was no molds observed for the cultivation of plain cracker and butter in PDA and NA, since there is no molds obtained in the incubation experiment. That's all from me. Thank you.
In conclusion, we can conclude that from 5 samples of food in closed system, rice, bread, and milk are the easier food for mold to growth. It is because bread has high moisture which allow mold to grow. On the surface of the bread sample, it had a small grey dot which is penicillium. For rice samples, it changed after 72 hours because of the condensation process that makes the water vapor trap inside the container. So it will change the temperature and the humidity on the container and it become pale yellow mold. For meat sample, it coagulated like curd and had a sour smell after 48 hours. It is because of the increase of lactic acid and the pH become more acidic. In an open system, rice and milk had a lot of growing molds. Meat sample changes after 48 hours by forming suspended white solid. For rice sample, it changed after 72 hours, which was it has an orange color, mold spot, and appears moist, soft, and yellowish color. For bread, it became dry and hard because it is lost moisture. The cracker samples for open system and closed system were in the same condition. The difference was that in the open system, it was soft and dry, meanwhile in closed system, it still appeared fresh. Butter does not have a mold because of the high fat content and relatively low water content. Growth media is needed to observe the microorganism on the solid agar.